Hi there, in this video we are going to discuss about receptor proteins, and then we will move on about nuclear receptor, then receptor without enzymatic activity, and then we will discuss adhesion receptors. So first we move on toward receptor proteins, and receptor proteins are basically proteins which are coupled to the receptor, and these receptors are basically G protein coupled receptors. It means that receptor cell surface receptor is coupled to G proteins. G proteins are of three kinds. These are GS protein, GI protein, and third one is GQ protein. Okay, so these three proteins are involved in different kind of signaling, but the basic mechanism of all three G proteins is same. A G protein has three subunits. One is alpha subunit, one is beta subunit, and one is gamma subunit. The alpha subunit is normally bound to GDP. GDP stands for guanosine diphosphate, and it is a nucleotide. Okay, so alpha subunit is normally bound to GDP, and when it is bound to GDP, it is inactive. This is inactive form. However, when a receptor is occupied some hormone, what happens? Alpha subunit releases its GDP and binds to GTP, and when it binds to GTP, G protein becomes active, and alpha subunit breaks away from beta and gamma subunit, and alpha subunit breaks away from beta and gamma subunit, and this alpha subunit is active. When it is bound to GTP, it is active, and it then moves on either to activate or to depress the cellular enzymes and leads to the cellular response. However, this alpha subunit also has intrinsic GTPase activity. It means that GTPase means it has enzymatic activity to break down this GTP. So transiently, after becoming active, this alpha subunit breaks down this GTP. One phosphate is removed, and GTP guanosine triphosphate gets converted to GDP or guanosine diphosphate. And when it converts to guanosine diphosphate, alpha subunit again becomes inactive. And when it becomes inactive, it again combines with beta and gamma subunit, and G protein again become inactive, and signaling cascade stops. So it is basically an overview of G protein. And G protein, all these G protein, GS, GI, and GQ show same behavior in cellular signaling. So we next move on to discuss them individually. Okay, so first we discuss the GS protein. Supposedly this is the cellular membrane here, and this is outside, this is inside of the cell. Okay, so what happens, suppose this is the receptor here, serpentine receptor. Supposedly it has 7 transmembrane helical structures. We know that protein exists in primary, secondary, tertiary structure. So it has a 7 membrane transmembrane protein segments which are in helical form. Okay, helical means like a spring. Okay, so outside on outer domain, here binds the hormone. Supposedly GS is usually pathway is followed by epinephrine. So we suppose that epinephrine binds here. It is bound by epinephrine hormone. So epinephrine binds here. So what happens on inner side? This is in association with G protein. Okay. So this is alpha subunit of G protein. This is beta subunit. This is gamma subunit, and it is alpha subunit is bound to GDP. It is bound to GDP. So when hormone binds here, this GDP is released, and it is replaced by GTP, and this alpha subunit moves on along the cellular membrane here, and this alpha subunit is now bound by GTP, and it is active. And when it is active, it activates cellular enzymes. And which enzyme it activates? It activates an enzyme which is located in cellular membrane, and that enzyme is adenyl cyclase. Adenyl cyclase enzyme. And what does this enzyme do? This protein activates this enzyme, and this enzyme converts ATP into the cyclic AMP. It converts ATP into the cyclic AMP, and this cyclic AMP in turn it activates other cellular enzyme. For example, it activates protein kinase A enzyme. Protein kinase A enzyme, and this protein kinase A, wherever the word kinase means, it means that this enzyme will be adding phosphate to some other proteins. So it will be doing phosphorylation. So this protein kinase A will phosphorylate other cellular enzymes and will lead to the cellular response. Okay, so it was the functioning of GS protein. GS protein is associated with receptors which are activated by epinephrine. For example, other hormones also work by this pathway. I am just taking an example of epinephrine. So epinephrine binds here, It activates G protein. G protein replaces GDP and binds to GTP. Alpha subunit along with GDP moves to activate adenyl cyclase enzyme. Adenyl cyclase converts ATP to cyclic AMP, and then cyclic AMP activates protein kinase A, and this protein kinase A activates other enzyme which lead to the cellular response. For example, this protein kinase activates an enzyme phosphorylase kinase B. This protein kinase A converts this phosphoryl kinase B from its inactive form to active form, and active form of phosphoryl kinase B converts glycogen phosphorylase from its inactive form to active form. So what does this glycogen phosphorylase do? It is involved in breakdown of glycogen. It breaks glycogen and increases the level of glucose in blood. That is an effect of epinephrine. So what has happened here? GS protein activated protein kinase A. In the end, this protein kinase A lead to the cellular responses. An example of cellular response is this protein kinase A activates an enzyme which is phosphoryl kinase B. This phosphoryl kinase B, when it becomes active, it activates another enzyme which is glycogen phosphorylase, and this glycogen phosphorylase lead to the phosphorylation of the glycogen and leads to the breakdown of glycogen, which lead to the increase in blood glucose level, which is an effect of epinephrine. So it is how this pathway lead to the cellular responses. Okay, now we move toward the functioning of GI protein. GI protein is basically inhibitory protein. It works in a very similar way. However, one key difference here is GS is stimulating adenyl cyclase. However, what does GI protein do? Supposedly here is the GI protein, here is the similar receptor here, and it is bound by some hormone, and here is GI protein. Okay, so alpha subunit of GI protein, when it bounds to GTP, alpha subunit after binding to GTP, it exerts negative effect or inhibits adenyl cyclase. So GS was activating adenyl cyclase, however GI protein inhibits adenyl cyclase. It works in same way alpha beta gamma subunit. When a hormone binds here, hormone activates GI protein, and GI protein in turn inhibits adenyl cyclase and leads to the decreased level of cyclic AMP. So basically GI protein effect is antagonist to GS. So GI leads to the decrease in cyclic AMP level. It is how GI protein is working. And which hormone works by GI protein? GI protein hormone is basically somatostatin. For example, somatostatin is growth hormone, inhibitory hormone. Growth hormone is released in response to hormone which is called growth hormone releasing hormone. This growth hormone releasing hormone comes from hypothalamus and acts on pituitary to release growth hormone, and it works by GS pathway by increasing cyclic AMP. So somatostatin is inhibitory and antagonist to this growth hormone releasing hormone. Then what does somatostatin do? Somatostatin works by GI pathway and it decreases cyclic AMP and leads to the decrease in secretion of growth hormone. So it is how GI protein is working. Okay, next we move on towards GQ protein, working of GQ protein. GQ protein is involved in the lipid metabolism. In the same way, here is the cellular membrane here, and here is the transmembrane receptor, 7 helical domains, and here outside it is receptor is bounded by some hormone. Here is G protein, alpha subunit, here is GDP, and when it bound to receptor G, alpha subunit becomes active, it moves on and it activates enzyme phospholipase C. It activates an enzyme which is phospholipase. And what does this phospholipase enzyme do? It cleaves another lipid. Here is a lipid in cell membrane which is phosphatidylinositol bisphosphate, PIP2. This is the structural lipid of the cell membrane, phosphatidylinositol bisphosphate. So GQ protein, alpha subunit of GQ protein activates phospholipase C, and this enzyme in turn break down phosphatidylinositol bisphosphate, which is a structural lipid of protein, into two compounds. One is PIP2, one is IP3 inositol triphosphate, and other one is DAG or diacyl glycerol. It breaks down into two compounds, diacyl glycerol and inositol triphosphate. So what happens? This inositol triphosphate goes to the endoplasmic reticulum and it binds to its receptor on endoplasmic reticulum and leads to the release of calcium from endoplasmic reticulum into the cytosol. And this calcium moves along with diacylglycerol and they activate an enzyme. They both activate an enzyme which is protein kinase. They activate an enzyme which is protein kinase C. They activate an enzyme protein kinase C, and this protein kinase C enzyme is involved in the cellular response. It phosphorylates cellular protein and leads to the cellular response. So how GQ protein has worked? When receptor is bounded by hormone, its alpha subunit becomes active, which activates phospholipase C enzyme. This phospholipase C enzyme breaks down the phosphatidylinositol into inositol triphosphate and diacylglycerol. Inositol triphosphate moves to the endoplasmic reticulum and leads to the release of calcium. This calcium, in association with diacylglycerol, moves to phosphoprotein kinase C, activates protein kinase C, and this protein kinase C in turn phosphorylates cellular proteins and lead to the cellular response. It's how GQ protein works, and this pathway is principally involved in the lipid metabolism. Okay. Next we move on about the another important signaling pathway, which is cyclic GMP or NO pathway, cyclic GMP and nitric oxide pathway. So what happens? This is basically shown in blood vessels and lead to the relaxation of blood vessels. NO pathway leads to the relaxation or vasodilation of blood vessels. So what happens? This is a endothelial cell. This is an endothelial cell, and this is a smooth muscle cell of blood vessel. So what happens? Parasympathetic stimulation, parasympathetic nervous system leads to the release of acetylcholine at the nerve ending near blood vessels. This acetylcholine comes to and binds to its receptor on the endothelial cell, and this receptor is not a ligand gated channel. It is not an acetylcholine ligand gated channel. Instead, it is a muscarinic receptor. It is a muscarinic receptor which is following. It is a muscarinic receptor which is following GQ protein signaling. Okay, muscarinic receptor are different from ligand gated channel. These are two main classes of receptor. One are nicotinic receptor and other are muscarinic receptor, which you will study in third year. You need not to go into details of these receptors. So when it binds to muscarinic receptor, it activates GQ protein, and in the same way, when GQ protein becomes active, here there will be activation of phospholipase C, and it will lead to the breakdown of PIP2 breakdown into the IP3 and diacylglycerol, and this IP3 will lead to the increase in calcium levels from calcium released from endoplasmic reticulum, as well as calcium will enter from the calcium channels here. So this calcium, what it will do? This calcium will bind to calmodulin, and calcium calmodulin complex will form. And this calcium calmodulin complex, what it will do? What has happened till now? Parasympathetic nervous system stimulation lead to the release of acetylcholine. Acetylcholine bound to the acetylcholine muscarinic receptor, which lead to the activation of phospholipase C. This phospholipase C lead to the breakdown of phosphatidylinositol bisphosphate into diacylglycerol and inositol triphosphate. Inositol triphosphate lead to the increase in calcium levels by releasing calcium from endoplasmic reticulum, as well as activating calcium channels. This calcium formulated calcium calmodulin complex. Okay, so no calcium calmodulin complex has formed here in a response to the acetylcholine. In response to acetylcholine, calcium calmodulin complex is here. This calcium calmodulin complex activates enzyme which is endothelial nitric oxide synthase. Enzyme endothelial nitric oxide synthase become active in endothelial cell, and it converts L-arginine, which is a amino acid here, into the L-citrulline, and in addition to this, nitric oxide is formed. So calcium calmodulin complex activated endothelial nitric oxide synthase, which lead to the formation of citrulline and nitric oxide from arginine. This nitric oxide diffuses to the smooth muscle cell of the blood vessel. It comes here and it activates an enzyme here which is guanyl cyclase. Guanyl cyclase enzyme become active here. This enzyme converts GTP into the cyclic GMP in the same way as previously ATP was converted into cyclic AMP. Here GTP is converted into cyclic GMP. So this cyclic GMP leads to the relaxation of blood vessel. How it leads to relaxation of blood vessel? It activates protein kinase G enzyme. This protein kinase G enzyme phosphorylates cellular proteins. It phosphorylates calcium channels. It phosphorylates lead to the decreased interaction of the myosin and actin chains and lead to the relaxation of blood vessels. So it's how this process is working on. Nitric oxide was synthesized from endothelial nitric oxide synthase. Nitric oxide diffused to the smooth muscle cell where it activated guanyl cyclase enzyme, which lead to the formation of cyclic GMP, and the cyclic GMP activated protein kinase G. This protein kinase G is responsible for the relaxation of blood vessels. So it's how this process is going on. So it was cyclic GMP NO pathway. Okay, here G protein coupled receptor, and here next we move on about the nuclear receptors. So how nuclear receptors work? Supposedly this is a cell here, and here is the nucleus. So nuclear receptor, for example thyroid hormone, in the blood they travel in combination to some binding protein. For example thyroxine, which is a thyroid hormone, travels by binding to thyroxine binding protein. In combination to their protein, they come to the target cell, and target protein remains outside and hormone enters into the cytosol. It moves to the nucleus, and in nucleus this hormone binds to its supposedly this is the receptor here, and it then interacts with DNA at specific nucleotide sequence, which are called hormone response elements. Specific nucleotide sequence are called hormone response element, where these hormones bind and show their response by altering gene expression. So it's how steroid hormones act by nuclear receptors. However, and an exception here is cortisol. Cortisol is also a steroid derivative. However, its receptor is present in the cytosol. Okay, here is its receptor. Cortisol came here, it binds to its receptor in cytosol, leads to the conformational change in receptor. Receptor releases its one molecule, which is chaperone. Chaperone is basically a protein molecule which maintains the shape of receptor. So this chaperone is released, then this hormone and receptor complex move to the nucleus, interacts with the DNA at hormone response elements and alters gene expression. So it's how steroid receptor work. Okay, so this was the working of nuclear receptor. Next we move on about the receptor without enzymatic activity. And these receptors include JAK-STAT pathway. We discussed the JAK-STAT pathway. So for JAK-STAT pathway, we take an existing cell membrane here. We take an example of leptin receptor. This is the receptor supposedly, and here comes the leptin and binds here to the receptor. Leptin bind to the receptor. After binding, this receptor undergo conformational change and dimerizes. It creates a dimer here. This is the dimer of leptin, dimer of receptor. Here is leptin, and this receptor associates with another protein and activates it in the cytosol, which is Janus kinase or JAK protein. JAK protein here becomes active. Okay, JAK protein, which is not a component of receptor, instead it is linked to it or attracted to it. This Janus kinase protein becomes active. It is first autophosphorylated, and then it phosphorylates the receptor as well, and it attracts other cytostolic protein. One prominent protein is STAT protein, signaling transducer and activator protein. STAT protein comes here, and this STAT protein is also phosphorylated by Janus kinase. It is phosphorylated and it is also phosphorylated by Janus kinase. Okay, so what has happened here? Receptor, there were two receptor, hormone bind here, receptor dimerized, and this dimerization lead to the this receptor binding and dimerization lead to the activation of Janus kinase, which was autophosphorylated and phosphorylated another protein, which is STAT protein. This STAT protein is now phosphorylated. It also creates a STAT dimer. It creates a STAT dimer. Here is phosphate and here is phosphate. This STAT dimer moves to the nucleus of cell, and interacts, it moves to the nucleus of cell and interacts with DNA at a hormone response element and lead to the cellular responses. So it's how JAK-STAT pathway is working. First there was hormone binding, then receptor dimerization, activation of Janus kinase, phosphorylation of STAT protein, dimerization of STAT protein, and then interaction with DNA and altered cellular responses. So it's how JAK-STAT pathway is going on, and this receptor intrinsically does not have enzymatic activity. Instead, enzymatic activity is shown by another protein, which is JAK protein, which is attracted to receptor, and this JAK protein is not a component of this receptor. So next we move on about the last receptor, which are adhesion receptors. Adhesion receptors are shown by WBCs. Supposedly this is the blood vessel, and here are endothelial cells. Okay, here are endothelial cells here, and here are the WBCs. So last adhesion receptor are basically selectins, and integrins. Selectins show weak association among the WBCs, and blood vessel and integrins show strong associations. So what happens? There are E-selectins which are present on endothelial cell. There are P-selectins which are also present on endothelial cell, and there are L-selectins which are present on WBCs. And WBCs show ligands for these selectin. Ligands means anything that bind to these receptors, which are usually formulated of sugars, and endothelial also show ligand for L-selectin. So these are selectin proteins are present on endothelial cells. Selectin protein also present on leukocytes, and their complementary ligands are present on endothelial cell as well as leukocyte. So they interact whenever there is cellular damage or damage to blood vessel. They interact, and weak interaction develop, due to which speed of the white blood cells slow down, and they attach to blood vessel, then break away, then attach, then break away due to this weak association. Similarly, WBCs also show integrins. They also show integrin protein, and their ligands are also present on the vessel wall. And when they interact, there is interaction of integrin and integrin ligands. A strong association develops, and leukocyte stops here. Leukocytes stop here, their movement stop. Then there are conformational changes in cytoskeleton, and they move out of the blood vessel by transmigration, and then move to the site of bacteria or tissue damage, and so on. It's how adhesion receptor work. So in adhesion receptor, principal protein are selectin and integrins. Selectin include P-selectin and E-selectin, which are present on endothelial cells, and L-selectin, which are present on WBCs. They show their complementary ligands, and they bound to their whenever there is activation of blood vessel or their tissue damage, they become active and bind to the their respective ligands, and then their association is weak. So blood vessel, so WBC speed slow down, they bind to blood vessel and then break away, then bind, then break away. However, when they integrins become active by chemokines and cytokines, there is strong association, and WBC stop here, and then they show cytoskeletal changes and transmigration from the blood vessel to the site of injury. So it was how different tissue receptors were working. So it was all about today's lecture. Thank you.